Humour as well then. Cillian de Gascun is a consultant virologist who graduated from Trinity College Dublin in 1998. He undertook his basic medical training in Dublin and Brisbane, returning to Dublin in 2002, and completed his specialist virology training in the National Virus Reference Lab at the University College Dublin and Barts and London NHS Foundation Trust in London. He's a Fellow of the Royal College of Physicians of Ireland, a Fellow of the Royal College of Pathologists in the UK in Virology, a Fellow of the Faculty of Pathology of the RCPI. His MD was awarded in 2011 by UCD for work carried out in the NVRL under the supervision of Professors William Hall and Colin Bergen. The primary focus of the MD was to describe the molecular epidemiology of HIV-1 in Ireland, where the work also documented the prevalence of antiretroviral transmitted drug resistance in Ireland over a 5-year period between 2004 to 2008. Following 2 years spent as a consultant virologist with Surrey Pathology Service in the UK, Cillian was appointed Director of the National Virus Reference Lab and returned to Ireland permanently in 2013. His main research interests continue to be in the area of molecular epidemiology of viruses, antiviral resistant, vaccine preventable infections, and the utilisation of these data to inform national health policy. Everybody will know, in January 2020, Cillian was asked to chair the SARS-CoV-2 Expert Advisor Group, a subgroup of the National Public Health Emergency Team. And we're delighted to have you back in Ireland, Cillian, and we're delighted to have you here today. So the stage is yours. Thank you very much.
Thanks, Amelia. Thanks for the kind introduction. Thanks for the invitation to speak here today. So hopefully my screen is sharing there. And when I first had a chat with the organisers about what to discuss today, obviously increasing laboratory capacity was very topical, but I suppose over the last few weeks and months we've learned a bit more about the virus, and I thought that increasing laboratory capacity might run the risk of putting your audience to sleep at the start of the day. So I will talk a little bit about increasing lab capacity, but I'm also going to talk something about the virus, given my own sort of virological background and the nature of the talks today. So let's see if I can move this on. Yeah, so everybody will be familiar with the background about SARS-CoV-2 at this point in time. So it was the WHO China country office that was first informed of cases of pneumonia of an unknown etiology in Wuhan city in Hubei province back at the end of 2019. And I think it's a really impressive sort of achievement of science that novel coronavirus, currently termed at that time termed 2019 nCoV, was announced as the causative agent by Chinese authorities only about a week later, on the 7th of January. And what's even more impressive from a laboratory perspective is that the first virus genome sequence was released for immediate public health support on the 10th of January. And then there were 4 other genomes deposited on the 12th of January in the virus sequence database, which is curated by the WHO's Global Initiative on Sharing All of Influenza Data. So it built on existing structures to allow the data to be shared. And the reason that was so important was because it allowed us to develop Christian Drosten's group in Germany, in collaboration with colleagues across Europe, to develop the first diagnostic assay within the first couple of weeks of the new year. So if we look at the sequence that we have from SARS-CoV-2, if you look at the phylogenetic tree on the left of the slide, at the very top in red, you'll see the novel coronavirus, now known as SARS-CoV-2. In orange, just beneath that, you'll see its closest bat precursor virus, RATG13, which is from Rhinolophus affinis bat species, and that was first isolated or retrieved in 2013 in Yunnan province. In the cyan colors, then you'll see a number of SARS-like sequences that have been retrieved from pangolins, and that led to an awful lot of speculation that pangolin might have been an intermediate host, analogous to the palm civets that we saw in 2003 with the original SARS virus. And then in blue, just below the cyan colored sequences, you can see the original SARS coronavirus from 2003. If you look down at the bottom half of the tree, then you can see our own seasonal human coronaviruses in green, which are scattered throughout the tree. And in essence, the fact that we could see the relationship between SARS-CoV-2 and the bat coronavirus really gave us an indication quite early on that this virus was likely to have come from bats, but the question was whether it moved directly or moved indirectly through an intermediate host. So just if we look at coronaviruses as a larger group, obviously we have our 4 seasonal human coronaviruses, NL63, 229E, OC43, and HKU1, and they're split between alpha and beta coronaviruses. And then the likes of SARS, MERS, and SARS-CoV-2 are beta coronaviruses. So there are 4 genera in total: alpha, beta, gamma, and delta. So one of the things I suppose that's intriguing for us as virologists is where this virus has come from. So if we look at the other coronaviruses that we've seen to date, they pretty much all either come from a bat reservoir or come from or transferred from rodents. Some have had an intermediate host that we've been able to identify, and some have had, we presume, have had an intermediate host, but we haven't yet been able to identify it. And I think what's important in the context of SARS-CoV-2 is that if we look at what has happened with all of the seasonal human coronaviruses that we have, the 4 ones that now cause the common cold, it's likely that when they cross the species barrier that they would have caused a more significant disease than they do at this point in time. And while that might give us some level of optimism that SARS-CoV-2 might mellow with age, that really is likely to happen over the course of years or decades, if not centuries. So it's not something that we can rely on in the short term. So I've highlighted a couple of the first points of this slide, why we believe bats to be the likely source, basically because since the original SARS coronavirus in 2003, a huge amount of surveillance has been done in bats, and we know that there are hundreds of coronaviruses circulating there. And the interesting thing about the 2013 bat virus that we've identified as the nearest relative, or the most similar sequence to SARS-CoV-2 at this point in time, is that despite being about 96% identical to SARS coronavirus 2 across its entire genome, the part of the genome that encodes for the protein that allows the virus to infect human cells efficiently, the receptor binding domain, or RBD, is actually quite different. And that receptor binding domain is actually far more similar to coronaviruses that had been identified in Malayan pangolins. And that's really where the speculation came from, that the pangolin might have been an intermediate source. And obviously we had the association with the live animal market in Wuhan back at the start of the pandemic. For those of you that don't know what a pangolin looks like, that's it. The meat is a delicacy in certain parts of the country, and the scales are used for inclusion in Western or Eastern medicines and various remedies. So it's one of the most trafficked animals in the world and is currently endangered, unfortunately. So it's getting its own back potentially. So the receptor binding domain is the most variable part of the coronavirus genome. And what we know about it is that there are 6 amino acids within that region that have been shown to be critical for binding to ACE2 receptors within the human body. So people will be familiar that that's how SARS-CoV-2 enters human cells at this point in time. And that really determines the host range of SARS coronavirus-like viruses, including SARS coronavirus type 2. And what's interesting for us at least is that the 5 of those 6 critical amino acids differ between SARS coronavirus 1 and type 2. So there was an awful lot of speculation at the start that this virus had been engineered somewhere, and that doesn't really stand up to scrutiny, purely because if you were likely to engineer it, you'd probably copy the original SARS coronavirus. And as I said, there's a significant difference within the receptor binding domain at those 6 critical amino acids. We do know that SARS coronavirus type 2 has a receptor binding domain that binds with high affinity to ACE2 from humans, from ferrets, from pigs, cats, and non-human primates. But as I said, the RBD sequence is different to that shown in SARS coronavirus type 1. So what this indicates to us about a potential source is that the coronavirus had time to evolve in the presence of a human or a human-like ACE2 receptor for natural selection. And as I said, it's unlikely that was in a pangolin. So what I want to highlight here is the receptor binding domain over on the left-hand side, and then I'll come to the polybasic cleavage site in a moment. So what I'm illustrating here is just a comparison of the sequence of the receptor binding domain and those 6 ACE2 contact residues. So at the top of the, so if we look to figure A here, at the top of that list of sequences is SARS-CoV-2. Then we've got the bat virus, or ATG13, followed by the pangolin, then the original human SARS coronavirus, and then some other bat SARS coronavirus related viruses. And you can see that across the 6 domains of the 6 amino acids that are boxed off here, you can see that there's perfect similarity between human SARS coronavirus type 2 and the pangolin RBD, but actually between the RATG13, they're quite different. And similarly, across the original SARS coronavirus, they're quite different. So what we can infer from this is that the available sequence data would suggest that the most likely divergence date of SARS coronavirus type 2 from its most closely related bat coronavirus probably happened somewhere between 40 and 70 years ago. And what this means is that viruses closely related to SARS coronavirus type 2 have actually been circulating unnoticed in horseshoe bats for many decades. And I guess the concerning thing for us is that this is now our third significant coronavirus emergence in the last 17 or 18 years. So given the high prevalence of these viruses in bats, it really means that these viruses are likely to cross the species boundaries again. Now, whether they will be as successful again remains to be seen. But if we look back over the last 3 that we've seen, SARS coronavirus type 2, or type 1 rather, managed to cross the species barrier. It managed to transmit from person to person, but it really only transmitted in the presence of symptoms, which meant it was relatively straightforward to control, but it also had a very high fatality rate. But it's no longer in circulation purely because isolation and quarantine worked. If we look at MERS coronavirus from 2012, again, it successfully crossed the species barrier and never really successfully managed to transmit from person to person. So we've still only had, since 2012, just on about 2,500 cases. That said, it hasn't gone away, and mortality rate is up around 30 to 35%. So very frightening if that were to acquire the ability to transmit more readily. And obviously SARS coronavirus type 2 has managed to successfully cross the species barrier. It's managed to transmit from person to person, and it's also managed to travel the world really in a few short months. So that really has hit the jackpot from a virus perspective, and there's no reason to think that it won't continue to stay with us unless we find some way of eradicating it. From a virus perspective, it's now identified a new host, and it seems to be perfectly happy with us at this point. Why has it been so successful? And I think this is sort of a question that can inform us for future study as well. So there's a polybasic cleavage site at the junction of the 2 subunits of the spike protein in SARS coronavirus type 2 that has not been observed in other lineage B beta coronaviruses. And what this does is that it allows for effective cleavage by furin and other proteases and plays a role in determining viral infectivity and host range. So the cleavage of that protein typically facilitates cell entry, and we've seen this in coronaviruses and also in influenza viruses. So basically furin is an enzyme that can cleave the protein, is abundant in the human respiratory tract, and is really found throughout the body. So as I said, in influenza viruses, acquisition of this polybasic cleavage site or a similar polybasic cleavage site can convert low pathogenicity avian influenza viruses into highly pathogenic forms. So again, if we look back at this, if we focus in on this previous slide and we look at this polybasic cleavage site, this is in essence a novel insertion that isn't seen in closely related viruses to SARS coronavirus type 2. So you can see there in RATG13 it's not present, in the pangolin it's not present, and in the original SARS coronavirus it's not present. And this is one of the issues, or one of the, I suppose, the key hallmarks of SARS coronavirus type 2 that has led to it being quite successful. The other interesting feature that we can take from this is that there's a leading protein inserted at that polybasic cleavage site, and this creates a turn in the protein that is predicted to result in the addition of O-linked glycans to the amino acids that flank that cleavage site. And again, this is unique to SARS coronavirus type 2. So O-linked glycosylation, and I'm sure many of you will be familiar with it and probably know more about it than I do, is in essence the attachment of a sugar molecule to the oxygen in the serine or threonine residues of a protein. And the function of this predicted O-linked glycans, it is unclear because we're still obviously learning about this virus, but it's potentially creating a mucin-like domain that's shielding virus epitopes or key residues on the spike protein. And several viruses we know utilize domains like this as glycan shields to evade an immune response. So what this tells us about, if we look at these O-linked glycans, what this suggests to us is that this virus, human SARS coronavirus type 2, has had the opportunity to evolve in the setting of an immune system because it's developed what could be seen to be an immune evasion method. So just to touch briefly on the theories of then where the virus may have come from. So obviously that it may have been the result of natural selection in an animal host before a zoonotic transfer. In support of this, we have the early association with the live animal market. We also have the pangolin coronaviruses that exhibit strong similarity to SARS coronavirus across the receptor binding domain. However, neither the bat nor the pangolin beta coronaviruses that we've sampled to date have the polybasic cleavage site insertion. Although it is seen in lineage A, for example, HKU1 has something similar. Furthermore, no animal coronavirus that is sufficiently similar to SARS coronavirus type 2 to date that could have served as the direct progenitor has been identified to date. But that said, we do know the diversity of coronaviruses in bats is massively undersampled. So for a precursor virus to have emerged, I suppose, de novo from an animal host, really the animal host would have needed a high population density and an ACE2 encoding region that was quite similar to humans to allow the receptor binding domain to evolve. Second possibility is that the virus emerged as a result of natural selection in humans following zoonotic transfer. So it's possible that the progenitor virus jumped into humans and acquired the genomic features that we discussed above through adaptation during undetected human-to-human transmission. And then once those features were acquired, it would have allowed the pandemic to take off and would have allowed for ready transmission of the virus from person to person. But based on the sequence data available to date, now obviously that's not perfect because there are viruses out there that have not yet been sequenced. We can infer that the RBD as described was present in a common ancestor. So that's based on the fact that we can see the receptor binding domain formation around those 6 key amino acids present in the pangolin. So obviously it's occurred in nature previously. So however, if we look at the polybasic cleavage site insertion, that appears to have occurred during human-to-human transmission because we can see that that isn't present in any of the most similar viruses to SARS coronavirus type 2 that are out there. So this could have happened if there had been many prior zoonotic events with short chains of human-to-human transmission. And for example, we have seen something similar with MERS coronavirus type 2, but it hasn't managed to adapt successfully to sustained transmission. And there are ways that we might be able to look back and see if this happened by studying banked human samples to see if there was an element of cryptic or undetected transmission from the animal reservoir into humans, and maybe with short chains of human transmission before the pandemic actually kicked off properly. And again, just to allude to this possibility of selection during passage, which was a theory that has been espoused, but there's no real evidence to support it. But at the same time, when a new virus emerges, there's no harm asking these questions and dealing with them reasonably. So basic research involving SARS coronavirus-like viruses has been going on really since 2003, and laboratory-acquired cases of SARS coronavirus has been reported. So it is an hypothesis that we can reasonably explore. So it is in theory possible that the receptor binding domain mutations were acquired in vitro, so in a laboratory setting, but that is unlikely given the fact that we know those mutations are present in viruses in nature. As I said, like the pangolin is the classic example, and they may be more widespread as well. But however, acquisition of the polybasic cleavage site does argue against a culture-based scenario because they really don't happen that readily within the laboratory setting or in vitro, although it has been observed in low pathogenic avian influenza viruses previously. In addition, we haven't really identified a progenitor virus. So again, you would imagine if somebody had identified a virus like this in recent years, it would have been published, it would have been reported, it would have been an interesting scientific finding. And furthermore, the generation of the predicted O glycans suggests the involvement of an immune system. And again, that doesn't realistically happen in the in vitro setting, so very unlikely. So I think we can dismiss that possibility. So at this stage, we still don't know definitively where the virus came from, but it's more likely that it probably transferred into humans first and had an opportunity to adapt there and acquire that polybasic cleavage site insertion. So why do I think this is important? Well, I think understanding the origin of this pandemic probably won't be able to prevent future zoonotic events, but it allows us to learn about what features viruses need to acquire to become more readily transmissible. Equally, if these specific mutations are required for a pandemic event to happen, then it's probably unlikely that they will happen by chance again in the future. Identifying the close relatives of SARS-CoV-2 in nature, again, should assist with studies of virus function. But at this point, we can't ascertain definitively how the cross-species transmission occurred. And I suppose the key issue there is that we don't know if the risk still remains. So are there animal markets either in China or around the world where animals with coronaviruses similar to SARS-CoV-2 are still being purchased, still being marketed, and still at risk of additional recombinogenic events? And that's the thing that coronaviruses can do. They're very large RNA genomes, and they recombine quite a lot. So we still can't answer that question as to whether there's an ongoing risk of further introductions into the human race. Increased surveillance obviously is vital, and to be fair to the Chinese, they've done a huge amount of surveillance over the last 17, 18 years, and so as we now know a huge amount more about coronaviruses in bats than we would have known previously. But again, if we could find a virus in nature that had an intermediate or a fully formed polybasic cleavage site in a similar virus as SARS coronavirus type 2, that would give us more support around the potential source and the natural selection hypothesis. And I suppose just to highlight that there's an awful lot of work ongoing in relation to trying to look at what viruses we have that are quite similar. And this is again another virus from the Yunnan province in China that was retrieved between in 2019, and this virus is actually more similar to SARS coronavirus type 2 across the length of the genome, but actually its receptor binding domain is completely different. So again, it does have a couple of insertions near the cleavage site of the spike protein, which again demonstrates that these things can occur in nature. I suppose one of the questions that I often get asked about is whether there are different subtypes of SARS coronavirus type 2, and anybody working in microbiology, be it in bacteriology or virology, will know that yes, subtypes happen and they emerge, and particularly with viruses, they can emerge quite quickly. Unfortunately, the academic community hasn't yet decided on a standard nomenclature scheme for naming these subtypes. So there are three proposals. GISAID on the right initially described different viruses based on particular mutations or particular polymorphisms at various locations. Then NextStrain, which was a superb online resource, tried to add some structure to that around the year of identification of the virus and then the particular subtype. And then Andrew Rambo and colleagues tried to put a little bit more structure that would align more closely to how we normally name different subtypes of viruses. So whether that's by letter or by number, and that lineage which he has nicely named the pangolin lineage is likely to become the most commonly used. But as I said, we'll see what's happened. So if we look at what's happening in Ireland, just for example, just to give you an example. So unfortunately, the pangolin lineages there, so pangolin stands for phylogenetic assignment of named global outbreak lineages. The key is in the center of that cladogram, but it possibly doesn't project terribly well. But you can see that even in a small country like Ireland, we have at least 7 or 8 different lineages there identified over at the, again, if you can look at that cladogram and look at about 9 o'clock on the picture or the image, you'll see 2 samples there, one from Cork and one from Dublin, and they're the very earliest sequences that we identified back in February and March that were very similar to the Wuhan strain originally. But then you can see we have obviously there's a huge number of lineages that have emerged since, and as I said, we see some geographic association between lineages and the place in the country. We can use these information or these data to identify related cases or perhaps specific outbreaks. But as I said, there is plenty of diversity in the country. At this stage, we've no indication that any lineage is more virulent or more severe from a pathogenicity perspective than any other. But as I said, there's always a risk or a concern that might happen. So just to go back to the laboratory side of this, as I said, the assay, thanks to the rapid release and sharing of genome sequence data, the assay was really developed in the first few weeks of January by Christian Drosten or Victor Corman and colleagues. And if you look just at the hour at the bottom of the slide, like the article was submitted on the 21st of January, that's 2 weeks after it's less than 2 weeks after the sequence was shared, and then it was published on the 23rd of January. So in Dublin here, we were able to start testing with the WHO recommended assay on the 7th of February. We had our first case then on the 29th of February. Just to flag the virology in the context of the structure of the virus and some of the proteins that we can target for the diagnostic side. Obviously, there's a spike, there's the membrane protein, there's the envelope glycoprotein, nucleocapsid protein, and then there's an RNA-dependent RNA polymerase in there as well. And if you look at what the first assay produced or developed was, it had an RNA-dependent RNA polymerase component, then the envelope and the nucleocapsid. And a lot of countries then, ideally, you didn't necessarily need to use all 3 targets, but certainly it was recommended that you would use 2. So we use the RNA-dependent RNA polymerase and the envelope back at that time. So shortly thereafter, commercial assays became available. And again, we switched to a commercial assay on March the 5th. And what this did was that it allowed us to increase capacity, because the initial laboratory developed test had probably a capacity of about 30 on a 96 well plate because of the different targets and controls, whereas the commercial assays then popped all of this into one cell. So all of a sudden you're getting 3 times the amount onto your 96 well plate. So I suppose just in the context of this talk, the capacity to, obviously at the moment people will be familiar with the fact that we're at 15,000 a day. We're above 15,000 a day now, but the capacity to do that scale of testing really didn't exist in Ireland back in February. People remember back to 2009 and the influenza pandemic. We tested an awful lot at the very start, but once the virus was circulating in the community, we moved to prioritising testing for the sickest patients and for hospitalised patients. And there was, back in February, March, there was discussion about whether you would do that with SARS coronavirus type 2 as well, whether you would move into a mitigation phase and just prioritise the testing of very sick and hospitalised patients. But it soon became, I suppose, quite apparent that this wasn't going to be another 2009, that we needed to test more widely. So back in February, realistically, you could have got to about 1,600 samples a day in the hospital system, and perhaps about 1,400 samples a day at the National Virus Reference Laboratory. And obviously at that point, there were no high throughput assays available. So the larger manufacturers like, say, Abbott, Roche or Hologic, they hadn't yet managed to develop their assays for their high throughput platforms. Similarly, building the capacity within our existing hospital system wasn't an option. Obviously, all of you are working in hospital laboratories, and you'll know that you didn't have spare high throughput capacity just sitting there waiting to be used, and 15,000 tests per day was no small ask, really, especially when there was a huge amount of pressure on trying to deliver it quickly. So the HSE did what made sense to do and engage with external partners in the public or private sector. And people did step into the breach. So the Department of Agriculture, Food and the Marine State Laboratory at Backweston, they had significant virology experience through their work with avian influenza and bovine viral diarrhea. And they also had available laboratory space. The one thing they probably didn't have was the experience of testing quite at that scale. And the Enfer group of laboratories out in Sallins and Naas, again, had significant virology experience because they are involved in the BVD eradication program, which, for those of you who aren't aware, involves the testing of all newborn calves every year, because BVD is like calves are infected in utero and then they shed virus chronically, and in essence, if the calves are positive, then they do need to be put down to protect the rest of the herd. But in essence, what Enfer Group had experience of was scale. So they were doing 1.4 million PCR tests per year. And maybe just to put these numbers in context, at the NVRL, in a regular influenza season, we would do about 15,000 influenza tests over the course of the season. So that's 15,000 between October and May. And what we're trying to do in this situation, obviously, is get to the setting of 15,000 a day. So it's unlike anything we'd ever tried to achieve before. But as I said, Enfer Group was in a position to provide support of that. Genomics Medicine Ireland, which is now Genuity, what they had the ability to do, and they were very helpful as well, was the ability to manufacture the WHO lab-developed tests at an industrial scale. So again, for those of you who are involved in making up lab-developed tests, obviously you've got to buy in your primers and probes and your enzymes and make up all of those things yourselves in the laboratory setting, but it's difficult enough to do that for a few hundred or a few thousand tests a week. But trying to bring it up to a situation where you can do 10 to 15,000 tests a day was very difficult. So GMI were very helpful in that regard. They weren't involved in any testing at that point. And then Eurofins by Omnis Again, most of you will be familiar with them. They're a medical diagnostic laboratory with an on-island presence, but they didn't have capacity on island to test at that point in time. But they did have access to an EU laboratory network, a European laboratory network, which is very helpful. So people will be familiar with all of the challenges that we had around the swabs, lysis buffer extraction kits, PCR kits, and of course, human resources, actually, people to do the testing and people to do the swabbing. All of the countries were looking for the same thing, obviously, in a pandemic, and these things weren't available. So we needed to build resilience into our system ourselves, and hospitals around the country were all pulling on the MagnaPure supply chain, for example, using it as an extraction platform because it's a very good platform. So what we have now is a variety of different platforms in different laboratories. Logistically, we were trying to keep the less sick people out of hospitals and away from GP surgeries for testing, and the National Ambulance Service stepped up and provided a home testing service, and then the community sampling hubs were established. And again, that was unprecedented in Ireland. We've never really had community sampling hubs set up in that way. Sample packaging and transport obviously was a consideration because it's a containment level 3 pathogen, which people don't necessarily have experience working with. And then communication of results in that third pathway, because when people attend their GPs or when people go to hospital, data flow is quite easy because we have health links, we have MediBridge, we have lots of ways of communicating results. But actually this was a brand new pathway which required new IT links and a new patient identifier. Now obviously it would have been easier had we our unique individual health identifier in place, but obviously we're not there yet. And then obviously setting up contact tracing centres. So we don't usually in the NBRL, I'm not sure about hospital laboratories, but we don't usually collect telephone numbers from our patients purely because we don't contact them directly. Our specimens come from GPs or hospitals, and they go back to the GP and to the hospital. We don't usually communicate with the patient directly. So therefore we had to create a new data field within the laboratory information system to allow it to accept mobile phone numbers, and obviously those things take time to develop. So just some of the challenges. And then obviously we wanted results to be able to go back to GPs, because patients obviously will call their GPs to discuss their cases, and if the GP doesn't have access to the result, but the problem was in the early days the GP wasn't actually involved in that care pathway, so which was very difficult again. So this led to a lot of, I suppose, a requirement for joined up thinking. In essence, there were 4 new systems. There was the electronic referral system, there were the sampling hubs with appointments and drive-in swabbing and specimen transport to the laboratories. There was a requirement for laboratory testing and the communication results, and then the contact tracing. And all of those, in essence, had to be set up from scratch and then linked. So the effectiveness and the efficiency was very dependent on the incoming demand and the capacity of that system. And again, people remember initially the indication for testing required a history of travel and also the presence of symptoms. And then that requirement for travel was removed. And I think that's probably when things really got serious, because again, people will probably recall the laboratory capacity was overwhelmed at one point in time over the course of a weekend. I think it was probably either just before or just after Patrick's Day. We got 50,000 referrals through the new electronic referral system from GPs over the course of 2 to 3 days. And at that stage, we didn't have the capacity to do that testing. So Eurofins by Omnis, in essence, came to our rescue, and they arranged for the testing of 30,000 samples overseas in Germany, which was fantastic. But it was a significant, I suppose, learning exercise for us that highlighted the need to ensure consistent capacity all through the system, and that there was no point bringing people into the system at the very start through the referral system, through the referral process, if you couldn't actually swab them in time and then get their specimens to the lab, and if you didn't have laboratory capacity to test them. And so the HSE has done a huge amount of work over the last 7 months to put in place an end-to-end system that is working really well for the vast majority of people. So I suppose my take-home message is, I suppose our current on-island testing capacity is about 100,000 a week, which is a really impressive achievement. It probably does need to increase further, and I think we saw that with a September surge after children went back to schools, that we, in essence, prior to that, we were seeing, I suppose, a referral rate of somewhere in the region of probably 10 to 12,000 per day, which was fine. But when schools went back, all of a sudden it was up above 50,000 and up above 15,000 a day, up to 20,000. So now that has plateaued and settled down again after the original return to school, but it certainly highlighted the fact that coming into the winter we'll probably need further capacity. But nonetheless, as I said, it's been a huge achievement, and just to acknowledge sort of the work of Paul Reed and Colm Henry in the HSE, and I suppose the unsung heroes within HSE procurement who managed to obtain all of the equipment and materials that we needed through that March, April, May period when it was a very competitive field, and in essence the supplies weren't out there. Like, we spoke to suppliers and they said their demand had gone up a thousandfold, or by 1,000%, and they just didn't have laboratory capacity, didn't have manufacturing capacity, because obviously nobody was envisaging this. Again, just to acknowledge, lab staff all over the country, none of these tests, again, there's sometimes a lack of understanding within the lay public who don't appreciate the amount of time and effort and work that goes into just, introducing new platforms, introducing new assays into diagnostic testing. So just to acknowledge the tremendous work done by laboratory staff all over the country, who really pulled together, and different assays were validated in different centres, and then all of those data were shared so that the country could sort of move forward as in solidarity. And again, we had huge support from academia and from industry, who stepped in and made up lysis buffer and tried to make up extraction kits, and were really very willing to. And they also provided their own staff who perhaps were furloughed from university and academic centres as they closed, and they went into hospital laboratories and started testing, and they went into swabbing centres and started swabbing. So it was a huge national effort. And I just specifically to acknowledge Mary. I know she's speaking later on, but Mary's done a huge amount of work in coordinating the laboratory response and all of the various platforms that go out to the various laboratories around the country, which make sure that, or will ensure that, we have resilience and robustness within the laboratory setting over this coming winter. And I suppose the final message is that in the middle of a pandemic is not a great time to start building laboratory capacity. So hopefully the infrastructure we're putting in place now will stand us in good stead when SARS coronavirus type 3 comes along in probably 5 to 10 years' time. Thank you very much.